Hi, this is Miss Litton, and this is my wonderful Period 1 AP Bio class. Say hi.
Hi.
And we just did a quick review to see what we remembered, and most of us remembered a lot, I think. This will be a way for you to fill in your notes. I'll go over that in your notes if you need a little bit of help, but take a look up here. Bacteria can contain DNA that protects them against, anybody know what this is from?
300.
Yes, it is. That can protect them against antibiotics. Okay, and keep in mind what antibiotics are there for. What are they trying to do? Kill bacteria, right? Anti-life. It usually kills it in one of two ways. It either an antibiotic prevents a cell from repairing its cell walls, or it doesn't allow it to make any more cells, any more cell walls. Either you can't repair it or you can't make any more. And so losing that cell wall is what's going to cause the death of that bacteria. So antibiotics, some bacteria are resistant to that. You know that, okay? So as long as they have those genes, then the bacteria will live. They can survive. This is a commonly used tool in genetic engineering because it's a way to select. Because whatever gene it is you're interested in, it's not like you're trying to make all these antibiotic-resistant bacteria, right? That doesn't sound like a wise thing to do. But it's a good way to select. If you put another gene of value to you right next to that gene, then if they have the resistance to the antibiotic, they also have the neighboring gene that you've attached to it. So that's one of the things we're going to look for in the lab. So here, for instance, is some E. coli. And it has the DNA that, if transcribed into mRNA and translated into a protein, will protect it against kanamycin. And this is one of the plasmids that you're going to be working with. Okay, is PCAN-R. It's got an R because it's actually a recombinant. It's been made. These plasmids are used as vectors, a way to take DNA from one source to another. And people buy plasmids based on recognition sequences they might have, or genes or antibiotic resistance that they want to have, and they can use it they can put their own genes in there and use that as a vehicle to transport that DNA. It's just like a thumb drive. And they can save things on this thumb drive, so they can put this thumb drive in this other computer and get this computer to generate whatever product it is you want. So this particular one has, and this is where, go into your notes on that one. I'll go into mine so I match you, potentially. Okay, so if you look at the plasmids, I'm going to show you the ORI sequence in just a minute, which initiates DNA replication. But go to where it says PCAN. Do you see where it says 5512? What do you think that stands for? Base pairs. 5,512 base pairs. So the CAN-R gene right here in yellow, what that's going to do is it's a gene that makes bacteria resistant to the antibiotic kanamycin. Okay, there's something else up here, PBAD. And PBAD, finish this line for me. RNA polymerase binds to the promoter. There you go. Okay, so PBAD is a promoter sequence for transcription of a specific gene. And also within here is another gene called RFP, and that stands for red fluorescent protein. So this is genes that code for red fluorescent protein. All right, now, you will receive little microfuge tubes of this pecan. And if you force, what is that called when you give bacteria DNA? It starts with a T. Not transcription. Transformation. If we transform, these are words you should know and love, if we transform this bacterium with this particular plasmid, if we try to kill it with the antibiotic kanamycin, we won't be able to do that. It will survive. Okay? A second plasmid that we're going to look at, okay, we look at this one, okay, is Piera. Is Piera or Piara bigger or smaller than pecan? How do you know that? Yeah, fewer base pairs. Okay, and if you look underneath it, AMP-R is a gene that makes the bacteria resistant to the antibiotic ampicillin. So you can see AMP-R right here in dark blue. Now they both have the ORI site, though they only drew it on this one. Could blue tell slate? It's up in your notes right there on the top. What's the ORI? What does that stand for? So the ORI site up at the top, what is that for? DNA what? DNA replication. Good. All right. And then there's another gene right here, ARA-C. ARA stands for a sugar, arabinose. And so ARA-C activates the neighboring promoter when bacteria is grown in the presence of arabinose. In arabinose. Basically acts like a switch. Now, do you remember how we said RNA polymerase binds to the promoter, but sometimes you might need assistance to help RNA bind to the promoter? Do you remember that? You need this thing activated to make PBAD be expressed. So in our last picture here, where we looked at this gene right here, this red fluorescent protein gene will never get expressed because there's no way to flip on the PBAD. So this gene right here is silent. You're not expressing it. And what you're really trying to do as genetic engineers is to cut this sequence out and go put it somewhere where we can get it expressed. You know where we could go put it? Yeah, go put it into our other plasmid, and once we do that, then we can turn the gene on with some arabinose and start making this red fluorescent protein. Now I'll explain that some more later. I'm just kind of opening your brain to that at this point. Okay, so our goal, here are your two plasmids, okay? So here's PCAN, and here's the PBAD and the RFP. Do you see where it says BAM in Hindi? BAM and Hindi are specific restriction enzymes. And those two restriction enzymes, remember what restriction enzymes do. They cut at a particular do you remember what it's called? Recognition sequence. So it's specific to that. So you're going to use I'll give you a mixture of those two restriction enzymes in there together. So you're just adding one solution, but it'll have both of them in there. And what we want to do is make some cuts right here around PBAD and RFP. So if we have a circle like this and we make one cut, that would be just still one fragment, right? But if we make two cuts, we'll have what? Two fragments, right? And we'll do the same thing over here at Piara, BAM and Hindi, cut. And then we'll have now a total, if we mix this, how many fragments will we have total mixing together? 4, right? 2 from each. And what we're hoping to do, okay, is to cut this out and sneak it in right over here. And if we can form this plasmid, okay, we'll form all kinds of plasmids. But if we can form this one, and we take this new plasmid and transform bacteria with it, and give that bacteria arabinose sugar to eat, do you see how we can turn on that gene? Then we can turn on that RFP gene. Okay? So when we do gel electrophoresis, okay, keep in mind, gel electrophoresis, we'll be able to verify, did we cut the plasmids? We'll also be able to see if we cut them in the right places. Because, let's go back and look at these sections right here. Okay, so this whole thing is 5,512 base pairs. Can I get eyes up here? This whole thing is 5,512 base pairs, and I remove PBAD. How many base pairs is PBAD? Oh, I'm so sorry. Let me go back. 807. So I can figure out how big this section is, right? How can I do that? Yeah, just subtract it off. So I know how big this piece is. Come over here, 4,872, and subtract off 377. I know how big each piece is. So remember, when you put DNA in a gel and the electric current goes on, which pieces move the fastest through the gel? The smallest pieces. So we should be able to predict a pattern that we should see if we take samples out of our cut plasmids, right? Should that look different than whole plasmids? Yeah, and that would be our control when we do our gel electrophoresis. We'll compare cut and uncut pecan, cut and uncut piara. And we'll also take a sample when we do our ligation, when we actually cut and then reform different combinations. We'll take a sample out of that tube too and do gel electrophoresis. And we should see all different combinations because we don't know what they're going to reform when we tape them back together. Because we can't say, no, only tape this. It's just going to tape anything where it matches a recognition sequence, it can tape it, right? And then we'll get all different combinations with that too. So we can see, did we cut the plasmid and did we cut it in the right place? We're going to use some very expensive marker. These are tubes of known size fragments of DNA that they make as a control, and we will let those run. It has 10 fragments in it, and we'll see where our different fragments line up based to that control, and we'll know if we cut it in the right places, and we'll know if we ligated it. Okay, so day one today of the lab is learning the tools, reviewing how to use gel electrophoresis and micropipetters. Then lab 2 is our cutting day. Lab 3 is our taping day. Lab 4 is our gel electrophoresis to check our work so far, right? And see if we cut in the right places, did we ligate? Day 5 is your God day when you transform the bacteria with your new genetically engineered DNA, okay? Then when we come back, will be our results. In this lab, okay, there are some bells we can't unring. Okay, so think this out. You're learning the tools today so you don't screw up. Next class period, when you cut, if you mix just one solution incorrectly, we won't know that till gel electrophoresis day 2 days later. 2 days later, when we do gel electrophoresis and we find out you screwed up, okay, that's an important point because after that, if you screwed up, which we find out 2 days later, you're going to be going around like this going, man, please have some of your DNA. And you're going to be begging from any leftovers from any other team. So do not underestimate the practice days because, you know, it's going to be literally over 48 hours before you know whether you made a mistake or not. Okay? So we have to be, I need you to pull out, you know, if you're taking OCD meds, stop. Okay? We need your full-on OCD to flare up. Start counting the tiles as you walk across the floor. Okay? Because you want to double check everything to make sure you're doing it correctly. Okay? I cannot underestimate that. Okay? I cannot overestimate that. Is that the right word? So when we do our gel electrophoresis, you will have a marker that we'll use to compare, and then we're going to have uncut and cut, and they should look very different from each other. And uncut and cut, again, this is of the piara, and then our ligated one should run pretty thick on the day that you do your checking. Okay, then our God day is our transformation day, where we force our bacteria to take in that plasmid that we made, and then we're going to plate it out on petri dishes and let it grow overnight in the incubator. Yeah?
Since it's like so small, the bacteria and the genes we're putting in, like how does the bacteria know to like take it?
It doesn't know. We're going to create a pressure differential having to do with temperatures, and that's a real specific thing where it's like got to be at this many degrees for this many seconds, and you pull it out and put it back on ice, and then you do this, and so it's like really specific, so you don't screw up on those days. Yeah. When you're God, you have to be, you know, really good about things. Yes.
So if we're putting the piara gene into the bacteria, doesn't that mean we're also making it resistant to ampicillin?
It does. Now, when it's not that we're adding the red fluorescent protein along with the P-bad into the piara, right? And that is one of the ways, because bacteria will grow on anything, so we will have controls when we plate out, and some will plate with no plasmids in them at all on straight up Luria broth, which every single bacteria loves, and they're like, I want to live here, okay? Anything would grow on it. And so you'll plate some like that, and then you'll plate some on antibiotics, and the ones on the antibiotics, it'll decrease the amount of growth you have, so only the ones you've transformed will live. And then you're going to have another plate that has antibiotics and arabinose sugar, and that one will be the only one that glows red. So when this lab was first started 25 plus years ago, when we first did this lab, the only thing we gave them was antibiotic resistance. That was the culminating part of the lab. And we did that for years. This now adding in, and I want you to think about the significance of this. This gene that you're going to use comes from a jellyfish. It's a mutated jellyfish gene. That's a eukaryotic gene that we're getting expressed in a prokaryotic cell. You see that? Not only are you playing God, but you're really mixing up like the species here. Do you see what I'm saying? That's huge. Now, who cares about making things red fluorescent, though you can make these RP glow fish that are kind of cool, right? But what would be really something you would want to make? Making insulin, right, for people? This is a way that you can just visibly see, because you will have bacteria that you're not going to give your plasmid to compared to ones you did give your plasmid to, and you'll see the difference phenotypically, right, in the proteins that express. You can visually see that, where it's kind of hard to look at a bacterium and go, I hope you're making insulin. Okay, you can't really see that. So that's why we're doing this with colors. Did that answer all your questions? Okay. All right, so let me get you caught up here on your notes part of it. Restriction enzyme, BAM and Hindi, each cut at different recognition sequences. Both plasmids have recognition sequences for the enzymes. And then I said, why 2 sites? And let me go back to that again. Why do we have 2 cutting plates? Why didn't we just use 1 restriction enzyme? What? I wanted to cut in 2 places, but again, why couldn't I just use the same restriction enzyme and just cut into, have it have 2 recognition sequences that are the same? What? Say it again? Yeah, I don't want it to recombine with itself, okay? Because I decrease the odds that it'll recombine with itself, and also the second reason is, remember when we talked about different ways DNA, remember we talked about additions and deletions, and we talked about inversions. We want the gene to go in, it needs to go in this direction. So we decrease the chances that it gets flipped in like an inversion by having it, because there's only one way to paste this in here. So it goes arabinose C, P bad, and then RFP. If we cut with the same restriction enzyme, it could have gone arabinose, RFP, and then P bad, and we wouldn't be able to turn that gene on. So we wanted it to go in a specific way. So that's the next part on your notes where it says, why 2 sites? It ensures that ligated DNA is oriented in the correct position. And it prevents the plasmid from reforming without the inserted gene. Okay, so lab 1, you're reviewing, learning the tools of, you're going to learn the micropipette and gel electrophoresis, just to review that today. Lab 2, you digest pecan and piara using the enzymes to what both plasmids? Cut both plasmids. Ligation, you're going to build piara R. This is our new plasmid. Combining fragments from pecan and piara to form a new plasmid. And then gel electrophoresis day is verification of digestion. And then lab 5, transformation, is getting the PRR into the bacteria to display the trait, that phenotype. Right? What will it look like? Something like this. Okay, so some of the bacteria here are glowing reddish pink and some aren't. Okay, this happens to be a plate that has the antibiotic ampicillin on it. So the only bacteria that survive on here have been transformed at least with the ampicillin gene, because you would need that to even survive on that plate. But these 2 colonies, keep in mind what a colony is. It's a single bacterium that then became what? 2 and then 4 and then 8. So these are great grandchildren, right? You think about it like that. These 2 colonies not only were transformed with that ampicillin gene, but they also had the RFP gene in it. And because this is a plate that contains arabinose sugar, it turned on the RFP gene, so it would express that trait. All right? Not it. Okay. One of you is going through the 5 steps of the lab in your own words. Try to put it in your own words. What's step 1, step 2, step 3? What are our 5 steps? Second bio buddy, explain why some of those are glowing and some aren't, and why there isn't solid bacteria on that plate. Go.
Number one, you're going to
And hope that's helpful.
Number three, you're like